Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we started discussing about immunology recently, where we discussed about immunity, the types of immune responses that we possess, and we also talked about the hematopoiesis, the process of formation of mature blood cells from hematopoietic stem cells. So we learned about how hematopoietic stem cells, which expresses CD34 as its surface marker, how it gets differentiated into certain progenitor cells, which are going to give rise to mature blood cells. So we learned about two lineages of blood cells, the myeloid lineage and the lymphoid lineage, right? So there we learned about dendritic cells, the type of cells which belong to both of those lineages. And today we are going to learn about dendritic cells. So just to summarize, when we were learning about hematopoiesis, we learned that hematopoietic stem cells, okay, they can give rise to two lineages, myeloid lineage as well as lymphoid lineage, right? So myeloid lineage, it can the myeloid progenitor cells, they can give rise to RBCs, okay, they can give rise to the granulocytes such as neutrophil, basophil, eosinophil, as well as they can also give rise to monocyte, which can be further developed into macrophages or dendritic cells. And myeloid cells, they can give they so this is how myeloid cells they can give rise to dendritic cells. Whereas the lymphoid cells, they can give rise to the B or T lymphocytes, natural killer cells, and dendritic cells. So today we are going to learn about dendritic cells, which belongs to the both lineages, the myeloid lineage and the lymphoid lineage. We have already discussed about all of the granulocytes, the neutrophil, basophil, and eosinophil, as well as the mast cells, which are the tissue resident granulocytes. Today we are going to learn about dendritic cells in detail, the location of dendritic cells in the body, the activation of dendritic cells, and what is the role of dendritic cell. That is what we are going to we are going to discuss in detail in today's lecture. So let's get started with the dendritic cells. So these are the master antigen presenting cells, means they are the ones which are going to present antigen to the other immune cells in our body. So these are the antigen presenting cells. I'll come to this point later, how they are going to present the antigen to the cells. Before that, let's learn about the history. So dendritic cells, these were first discovered by Ralph Steelman. Okay. When this researcher discovered dendritic cells, he got Nobel Prize posthumously after the discovery of dendritic cells. So he characterized the dendritic cells and studied the dendritic cells. So the dendritic cell, this word dendritic cell, it is derived from a word known as dendrion, which means tree branches. Okay, so it was named dendritic cells. Dendritic cells, they were named after this particular word dendrion, which has a meaning tree branches, because those cells, when viewed under microscope, they have a lot of projections, and those projections are helpful for antigen presentation. So actually, when Paul Langerhans, he was studying those dendritic cells, he found a few cells in the epidermis, okay, and he thought that those are the nerve cells. Okay, so because of the long projections of the dendritic cells, it resembled to the dendrites of the neurons, and that is why Paul Langerhans he thought that he mistook those cells for the nerve cells of epidermis, and that is why the name dendritic cells it gives us an idea about the morphology of those cells. So those cells have multiple. So these cells have multiple projection. Okay, something like this. This is how this cell looks like. Okay, so this is a bit exaggeration of projection. So this is somewhat it looks like. So it has structures like dendrites, and that is why it is named as dendritic cell. So those long membranous structure, those projections, they are covered with those projections. They are covered with membranous extensions. Okay, and they resemble to the dendrites of the nerve cell, and that is why they can also extend and retract according to the conditions. Okay, so they are dynamic in nature. So the projections of the dendritic cells, they are dynamic in nature because they can retract and extend according to the cell need. Okay, so those dendritic cells, they are antigen presenting cells. Well, we have other cells as well which do this job. They are macrophages and the B cells. Both of them are the antigen presenting cells. So alongside macrophages and B cells, dendritic cells also does this job. And the main cells who does this job are the dendritic cells, and that is why they are also known as master antigen presenting cells. So they belong to both myeloid and lymphoid lineage. And as they are going to, do this job of presenting antigen, they're also known as patrolling car. Okay, they resemble to patrolling car because the way the patrolling car has a job to roam around and to look for any suspicious activity, in the same way dendritic cells, they roam around in the peripheral tissue and look for whether there's whether there's tissue damage or whether there's infection. So they do dendritic cells, they constantly, they are constantly sampling the tissues for any suspicious activities such as the tissue damage, right, or the infection, right. So they look out for those activities, and they look out for the signs of infection, signs of tissue damage, and once they detect those tissue damage, they get activated. So upon detection of tissue damage or infection, those cells get activated, and once they get activated, due to the activation, they get mature. Okay, so the maturation start, and once they get matured, they express the surface markers that are required for initiation of immune response. So maturation or activation in this case, in the case of dendritic cell, means the expression of surface markers, means the receptors that are present on the surface, which are essential for initiation of immune response. Right, that this is how the initiating immune response is also one of the roles of the dendritic cells. So once they get matured and they get activated, those dendritic cells, they are going to report to the headquarters, means patrolling cars, the cops, or the patrolling people, when they roam around and they look out for the some suspicious activity. Once they have detected that suspicious activity, they have their own weapons as well, and then they are going to they are going to call for the other people for help. So other units, other troops for the help, right. So they'll call out reinforcements so that whatever situation is there, it is taken care of, right. So the patrolling cars, they are going to report to the headquarters for those suspicious activity. In the same way, dendritic cells, what do? They just migrate to the nearby lymph nodes, to the nearby lymph vesicles. So they drain to there, and then they present those antigens to the nice T cells, and that is what we call as reporting to headquarters. So those patrolling cars, means those dendritic cells, they also report to the headquarters, means They drain into the nearby lymph node, so they migrate to the nearest lymph node, and once they migrate to the nearest lymph node, what do? They present the antigens that they have collected from the site of infection. Okay, they present the antigens to the naive T cells. Okay, so what are naive T cells? These are the T cells which has never seen, which have never interacted to any antigens. So these are called as naive T cells. So naive literally means here innocent. So those T cells are immature and they haven't. So they are mature T cell well, but they haven't interacted with the antigen yet. So due to the antigen presentation by the dendritic cells, naive T cells get they get activated, and now they are going to show some immune response. So this antigen presentation plays a very important role for initiating immune response, and this is how the dendritic cells they are a part of link between the innate immunity and adaptive immunity, because T cells they are the part of adaptive immunity, and this is how for the initiation of adaptive immune responses, dendritic cells play a very important role. So the suspicious activity, whatever they have seen, means the antigens they have gathered at the site of infection. They are presenting them on a platter to the naive T cells, and this is how naive T cells are going to get an information about the suspicious activity or the damaging activity going on at the site of infection. Okay, so this is how the initiation of the adaptive immune response due to the antigen presentation will be done by the dendritic cells. Okay, so those dendritic cells they are localized in ubiquitously in all types of tissue, but they are positioned very strategically. So the way patrolling cars, they roam around the areas where the protection is required, where the public safety is required, right? So they patrol around the government buildings, they patrol around the some companies, important companies where the protection is needed, or the areas where the chances of where the chances of crime are high. So patrolling cars, they are positioned very strategically according to the need. In the same way, those dendritic cells, they are also strategically positioned in our body. So they are ubiquitously present, but wherever the location is there in the peripheral tissue, that is very strategic. So they are present in blood, obviously, but they are rare in blood compared to the other types of immune cells such as neutrophils. They are also present in skin. So the dendritic cells which are present in the skin, they are known as Langerhans cells. Okay, because they were discovered by a scientist, by a researcher, Paul Langerhans. Okay, these are known as Langerhans cells, and those Langerhans cells they were mistaken by Paul Langerhans as the nerve cells of epidermis. So they are found there. Then the dendritic cells are also found at the gastrointestinal tract. Okay, they are also found in spleen. They are also found in lymph nodes, and they are also found in the thymus. Okay, so these are the various tissues where those dendritic cells are located. So the dendritic cells they also have a capacity of phagocytosis. So they are also phagocytic cells. Okay, but they have less phagocytosis or less phagocytic activities compared to the macrophages. Compared macrophages. So, okay, and compare if we compare them to macrophages, the hydrolytic enzymes they will also be less. They have less hydrolytic enzyme compared to macrophages, as well as few digestive bodies such as lysosomes. Okay, they are few in number compared to the macrophages, because the phagocytosis is not the it's not the primary function of the dendritic cells. Dendritic cells they are only alarming our immune system against the infection, against the pathogen. They also play a role in in the tolerance, means the recognition of self cells apart apart from the non self cells. So they have, they serve those antigens on the platter to the, to the other immune cells, and it's like, look what I've found. And now you decide whether this is harmful for our body or not. So they are kind of the cells which are going to help in tolerance. Tolerance is the, it is the characteristics that is possessed by our immune system, means the characteristic of our immune system to be able to differentiate between the self cell and the non-self cells, right? So non-self cells, they are, they are viewed, they are visualized as a threat to our body, where self cells are our own cells, right? Our own cells are, they are our cells, and that is why we don't need to eliminate them. They are not harmful, they are not damaging to our body, and that is why the distinguishing between self cells and non-self cells, it is very crucial, and that ability is the one of the abilities, one of the characteristics that our immune system possesses. And for this tolerance capacity, the dendritic cells also help the immune cells. So for the tolerance, dendritic cells also play an important role. So dendritic cells, they play important role in patrolling. So looking out for some suspicious activity. They also play a role in initiation of immune adaptive immune response, and they also play a role in tolerance. Okay. So when we have to dendritic cells, as I told you, they are very rare in the blood circulation. So when we have to do some in vitro experiments and we have to generate dendritic cells in vitro, we use the chemical signaling molecule GM-CSF, which is actually a cytokine. So this is a colony stimulating factor, and I will discuss about this GM-CSF in detail later in the, in the cell culture series. So well, GM-CSF is granulocyte macrophage colony stimulating factor. Okay, it is granulocyte macrophage colony stimulating factor. So this GM-CSF, it is very important. When we use GM-CSF, then we can, generate the dendritic cells in vitro. So this is used for in vitro generation of dendritic cells. Okay, because for the development of dendritic cells in vitro, GM-CSF can work wonders. Okay, it also works for, it also enhances the phagocytic, the engulfment capacity of the granulocytes and macrophages. So for enhancing, for making them efficient, this colony stimulating factor is also used, and that is why it is named as GM-CSF. Okay, so this is how we can obtain the dendritic cells in vitro in the lab settings, and we can use them for various experiments. Okay, so because the viability of dendritic cells is very, it is very poor, and again they are very rare in the blood circulation, and that is why it becomes difficult to extract them and to carry out any type of experiments. So the poor viability as well as the rarity, both of them are the challenges, and that is why we use this GM-CSF, this factor, this cytokine, this signaling molecule, for in vitro generation of the dendritic cells. Okay, so this is the kind of introduction about the dendritic cells. Let's now have a look at activation and the antigen presentation by the dendritic cells. So let's now take a look that how those antigen presenting cells, those dendritic cells, they are going to get activated and they are going to do the antigen presentation. Okay, so so once there are, antigens present inside the body. Well, I did not discuss this before, so let me discuss a few terminologies. Antigen, okay, versus immunogen. So whenever in our body any foreign substance is entered, that foreign substance is referred as antigen. Whenever there's some lipid molecule, sugar or protein, no matter what type of chemical composition that substance has, but any type of foreign substance when it enters in our body, that is referred as antigen. And if this antigen, means this foreign substance, is able to evoke the immune responses, then it is referred as immunogen. Okay, so antigen is any foreign substance that is entering in our body, while immunogen is the foreign substance that is capable of inducing immune responses. Okay, not all of the all of the foreign substances that enter in our body, they evoke immune responses. For example, dust. We take a lot of dust, a lot of bacteria daily in our in our system, but it's not necessary that those particles, not the bacteria, but those dust particles and all of those stuff is going to evoke any immune response. So such foreign substances that are taken in by our body, they're referred as antigens. And the if they are evoking any immune response, then they are called as immunogens. But sometimes those factors, they causes allergic responses, means the inflammatory responses, where our immune system is working, but it is not actually eliminating the pathogen, but it is just showing inflammatory immune responses. So many times antigen, it causes allergy, such as pollens, such as dust. A few people have dust allergy, right? While others don't have any type of effect by the intake of the dust. So whenever those antigens, they are causing the allergy in case of humans, in case of individual, then they are referred as allergens. Okay, so I wanted to discuss this before we proceed further. So I hope the difference between antigen, immunogen, and allergen is clear to you all. So let's get back to our topic, how the activation and antigen presentation is done by dendritic cells. So whenever antigen enters in a body, that antigen will be recognized, it will be taken up by the antigen presenting cell, dendritic cell. So whenever there's the physical barrier, or the chemical or the biological barrier has been invaded by the has been evaded by the pathogen. So now this pathogen has crossed, physical, chemical, or biological barrier that our body had, and now it is going to face the dendritic cells. So it is going to face the innate immune system in our body, and that is dendritic cells. So what does dendritic cells do? They take in those antigens that are present in that inflamed tissue, in that infected tissue, by either phagocytosis, receptor-mediated endocytosis, or macropinocytosis. So the very first response by the dendritic cell is the antigen uptake. So they simply take in the antigens inside them. So the antigen uptake that can be seen by either phagocytosis or the receptor-mediated endocytosis or micropinocytosis. I haven't discussed about pinocytosis yet, and I haven't even discussed about the mechanism of endocytosis, exocytosis. We'll discuss about all of this stuff in the cell biology 201, right? We have covered 101 cell biology. We haven't yet discussed about 201. So we'll discuss this all of the mechanisms of those phenomena in that lectures. But for now, the pinocytosis means the cell drinking. It is also referred as cell drinking, when the cell from the extracellular cell engulfs the extracellular fluid in such a way that this extracellular fluid will also be transferred inside the system, inside the cell, as well as the substances which are dissolved in that extracellular fluids that will also be taken inside the cell. So phagocytosis, if phagocytosis is cell eating, okay, means cell is eating some extracellular substances, then pinocytosis is referred as cell drinking. Okay, so for now, knowing this much is enough. So either by phagocytosis, receptor-mediated endocytosis, or micropinocytosis, the antigen will be taken in by the dendritic cells. And now once this antigen is taken in by the dendritic cells, they are going to induce the innate responses, as well as they are going to present this antigen to the adaptive immunity. So the next thing that is done by the dendritic cell is to inducing immune responses, innate immune responses first. Okay, means it is going to release cytokines that will evade other immune cells to accumulate at the area of infection, and that is going to result into the initiation of the innate immune responses, right? So the dendritic cells, they are going to release cytokines. Those are the chemical signals. These are the immune system mediators, right? So they are going to alert other immune cells for the activation of immune responses. And dendritic cells, they will also present antigen, so that the antigen which they have taken in, that will be presented. So the presenting antigen to the T cells. Okay. So the antigen presentation to the T cells, which are the part of adaptive immunity, that will be done with the help of MHC molecules. So with the help of MHC molecules, the antigen will be presented to the T cells. Okay, depending upon, so there are different classes of MHC molecules. Well, MHC molecules, they are major histocompatibility complexes. Means they are the group of proteins which are important for the antigen presentation, and they are the ones which are going to tell us about the compatibility of the tissue. So they are going to decide the compatibility between the two tissues. So what happens is that all of the cells, so there are two classes of MHC molecules. Well, I am going to discuss about MHC molecules in detail in some separate lecture, but for a kind of introduction, learn this, that there are two types of MHC molecule, the class 1 MHC molecule and class 2 MHC molecule. So the class 1 MHC molecule, they are present in all of the cells that are present in our body, except the ones which doesn't have nucleus. This is present on all of the nucleated cells. Okay, this means RBCs doesn't have this MHC molecule, whereas the MHC2 molecules, they are only present in the antigen presenting cells. Means they are present over the dendritic cells, the macrophages and B cells. So only dendritic cells, macrophages and B cells are the ones which are going to have MHC2 molecules. That is why the dendritic cells, macrophages and B cells, they have nucleus. This means those three types of cells will have both MHC1 and MHC class 2 molecule, but all of the other cells except RBC, okay, except RBC, all of the other cells that are there in our body will have MHC1 molecule. And this is why the MHC molecules, the major histocompatibility complex, they are also referred as HLA. Okay, so this is HLA typing is very important for in the cases of tissue grafting, when you have to transfer a tissue, when you have learned, you have seen this in movies. Whenever there's tissue transplantation, first compatibility check is very important. So whether the tissue is going to be compatible with the donor tissue is compatible with the acceptor person, with the receiver, that is important. So that is why we do the HLA typing with this MHC molecule. They are also referred as HLA, okay, human leukocyte antigen. So the HLA typing means the matching of the HLA types. It is very important in the case of tissue grafting, in the case of any tissue any transplantation surgeries. So that is why whenever we there's blood transfusions, we only check for the blood types. Why the any why the any person can donate the blood? Because the blood cells do not have MHC1 molecules, and the MHC1 molecules, they are HLS, means human leukocyte antigens, means they are the flags present on the cells which are which are screening that this cells belong to this person A. Okay, so the person B will have different types of MHC1 molecules. So those are the little flags which are present on all type of cells, which are labeling those cells that they are owned by this person. They are owned by this particular individual. That is why whenever cells from other person they are infused, they are introduced in the body of the body of a receiver, which has different types of MHC molecules, the body will reject those cells and they will kill those cells. And that is why the HLA typing means matching, seeing whether those molecules are compatible or not, whether they are similar or not. It is very important in the transplantation surgeries. Okay, so that is why RBCs, which does not have MHC1 molecules, if the blood group of those two persons, means the antigens that are present on the RBCs, that matches, then we can easily, any person can donate his or her blood to any person, right? Because RBCs doesn't have this MHC1 molecules, and that is why they won't be rejected by the system, by the receiver, by the immune system of the receiver, uh, by differentiating them as non-self if the blood type is self. Okay, if I have blood type O positive, and some other person has donated his blood O positive to me, my body won't reject that O positive blood, because it will recognize that O positive RBCs as its own. But when it is done for any other tissue, for heart or for any other cells, that those cells will be rejected by the immune system of an individual, by the host, considering that those are the non-selves. So that this is how the MHC molecules, which are present on the dendritic cells, on the macrophages and the B cells. So in the case of dendritic cells, those MHC molecules, they are also helpful for tolerance. This is how they are going to differentiate between self and non-self. So dendritic cells, they have both type of MHC molecule class, the class 1 and class 2. Macrophages and the B cells, they also have both of the MHC class 1 and class 2 molecules. But if the question is, what type of MHC molecules does neutrophils have? So they only have MHC type 1. Okay, so MHC class 1 molecules. So those MHC molecules are the ones which are going to represent those antigens that are taken up by that taken in by the dendritic cells to the T cells. Okay. So though they are the ones which are going to do the antigen presentations. So those if the MHC type 1 it is going to it is going to present the antigen, then the MHC type 1 will present its antigen to the cytotoxic cells, cytotoxic T cells, means CD8 positive T cells. Okay. Well, if the MHC class 2 molecules are the ones which are going to represent the antigens to the T cells, then they are going to represent it to the CD4 positive T cells. Okay, I haven't discussed about T cells yet, and that is why if you don't get what are CD8 positive T cells, means the cytotoxic T cells, or the CD4 positive T cells, means helper cells, if you don't know about them, don't worry, we'll discuss about this later. So for now, it's important to know that MHC1 molecule, they are going to represent whatever is going on inside the cell to the immune cells. On the contrary, MHC2 molecules, they will present whatever going outside in the environment to the immune cells. Okay, so let's assume that this is the cell which has both MHC1 molecule and MHC2 molecule. So this cell has both MHC1 molecule and MHC2. So this is MHC type 2, and this is class 1 molecules. So the class 1 molecule, so there will be some antigens present inside the cell that belongs to this particular cells, and there will be some antigens that are present outside the cells. Okay, maybe they could be maybe some viral antigens or the bacterial antigens. Again there could be some antigens that are taken in by the cell. If the virus has infected, then cell can also, those molecules can enter either inside the cell, or they can even stay in the extracellular environment, right? So type 1 MHC molecule, they will be presenting those the antigens which are present inside to the to the immune system cells. Okay, whereas the antigens which are present outside the cell, that will be presented by the type 2 MHC molecules to the immune cells. Okay, so this is again other difference between type 1 and type 2 MHC molecules. Okay, so this is how antigen presentation will be done by by those dendritic cells. Okay, so there are different types of dendritic cells because dendritic cells they have different different morphology. So there are different types of dendritic cells depending upon function or depending upon morphology. So those types of one of them is the plasmacytoid dendritic cells, okay, which are written as PDCs. So this plasmacytoid dendritic cells, they are named plasmacytoid because they, resemble to the plasma cells morphologically. So they resemble to the plasma cells. So plasma cells are the antibody producing cells. So they resemble to those cells morphologically, and that is why they were named as plasmacytoid dendritic cells. Okay. So those plasmacytoid dendritic cells, they are very important. They play a very important role whenever there is viral infection. Okay, so during the viral infection, those dendritic cells they play antiviral role. How they secrete the interferons, type 1 interferons. So they secrete type 1 interferons which are important, which has antiviral properties. So these type 1 interferons, okay, type 1, oh, just a second. Okay. So type 1 interferon, these have, okay, these have antiviral properties. So they are effective against the viral infections. So the plasmacytoid dendritic cells, they secrete a robust amount of type 1 interferons that will be helpful against the viral infection. And this, so that the I interferon alpha, interferon gamma, beta, they will be secreted by those plasmacytoid dendritic cells, and this is how those plasmacytoid dendritic cells they are going to play a role in the initiating innate immune responses, because interferons they are a part of innate immune responses. So this is how the plasmacytoid dendritic cells they have antiviral property, but alongside antiviral property they also possess antitumoral property. So they are also antitumoral in nature. Okay, this has been seen in in many lab experiments. So plasmacytoid dendritic cells, they initiate the innate immune responses by secreting interferon, as well as they also do the NK cell activation. Okay, so they are also important for natural killer cell activations, and they are going to take part in innate immune responses, but they are very poor antigen presenters. Okay, they are quite poor antigen presenters. So the dendritic cells, when they are in their immature form, they they are more of phagocytic in nature. They are highly phagocytic, but once they get mature, they get more specialized for the antigen presentation. Okay, so this is a very important point to note down, that whenever the cells, the dendritic cells, they are in their immature form. Okay, so at their immature form, the priority or the working mechanism, the priority will be given to phagocytosis. So they are phagocytic in nature. They are highly phagocytic, but they are very poor antigen presenters. So they are poor in the T cell activation. Okay, so once those immature dendritic cells they get mature, now the focus they get more, they get differentiated, and they get more structured, sophisticated for the antigen presentation. So once they get mature, they are now more likely to be good antigen presenters. So they will be, they will be trained now for antigen presentation. and the phagocytic activity, which was quite high before, that will decrease. So the phagocytic activity will decrease, and that is why dendritic cells, they are less phagocytic in nature compared to macrophages. So this transition occurs when the dendritic cells, they get matured. Okay, so this is what we can see when during the maturation of the dendritic cells. And that is why the different types of plasmacytoid dendritic cells, they also focus on different types of function. For example, the plasmacytoid dendritic cells, they focus on the generation of innate immune responses, the antiviral innate immune responses such as generation of interferon alpha and beta, and they also activate the natural killer cells. But they are poor antigen presenters. So the more focus is on innate immune responses rather than on the adaptive immune responses, because due to the antigen presentation, adaptive immune responses, they are getting initiated. So that's why we can say that plasmacytoid dendritic cells, they are more focused on the generation of innate immune responses. Then there is other type of dendritic cells which are actually not a part of immune system, but they are also important and they have completely different role. So as of now, I told you that dendritic cells, they have a role in activation of innate immune responses or activation of adaptive immune responses. Innate immune responses will be generated due to the release of cytokines or release of interferons, whereas the adaptive immune responses will be generated due to the activation of T cells. Right? So that is the main job of dendritic cells, presenting antigens to the adaptive immune cells or generating the innate immune responses. So that is why they are referred as antigen presenting cells. But there's a type, if I tell you that there is a type known as follicular dendritic cells, because they are present in the structures of lymph nodes called lymphoid follicles. Okay, so they are, they are very abundant in lymphoid follicles. So those lymphoid follicles has those follicular dendritic, those dendritic cells, and that is why they are named as follicular dendritic cells. So those dendritic cells, they are not antigen presenting cells, rather than they are very important in the B cell biology. Okay, so these are very special. So rather than calling them antigen presenting cells, we call them specialized antigen presenting cells, and they play important role in B cell biology. So what do? They are actually helpful in the development of B cell, in the differentiation and the maintenance of B cells. So they are important in development, differentiation, as well as maintenance of the B cells. This is how they are different, they are distinct compared to other dendritic cells. So besides the plasmacytoid dendritic cells and the follicular dendritic cells, we have other type which are also known as conventional dendritic cells, or the ones which originate from the myeloid progenitor. So that is why they are known as myeloid dendritic cells, because they belong to the myeloid lineages, and that is why they are also written as MDCs. MDCs. Okay, so those are MDCs, the myeloid dendritic cells. They are also classified into CDC1 and CDC2. We are not going to discuss this in detail. Well, there was CDC1, those dendritic cell. They activate the CD8 positive cells with the help of MHC type 1 molecules, and CDC2, those dendritic cells are going to activate CD4 positive T cells with the help of MHC molecule type 2. So those myeloid dendritic cells are there. Then there are the Langerhans cells, which are the dendritic cells of skin cells. Then there's other type of dendritic cells which are which are present, which are differentiated from the myeloid from the monocytes. So you remember here when we discussed about hematopoiesis, the granulocyte monocyte progenitor is going to give rise to monocytes, and it can all those monocytes can be, can give rise to macrophages, but they can also give rise to the dendritic cells. So there exists a different type of dendritic cells which are, monocyte derived dendritic cells. So these are the four different types: the plasmacytoid dendritic cells, the follicular dendritic cells, as well as the myeloid dendritic cells and the monocyte derived dendritic cells. So with this, I'm done with the lecture. I have discussed about the dendritic cells in detail. We have discussed about the roles of dendritic cells, how they resemble to the nerve cells of our body due to their dendritic structure, due to their dendrionic structure, which is a which are the long projections present, which are dynamic, which can extend and retract, and how the antigen presentation is done by the dendritic cells. We learned about that. So with this, I'm done with this lecture, dendritic cells. In the next lecture, we'll discuss about macrophages, and then we'll start discussing about natural killer cells. And once we're done with the cells of immune system, we will also discuss about the organs of immune systems. So till then, see you. Bye bye.